Okay, we're getting to the end here. So on our phylogenetic trees, we need to talk a little bit about time and how time passes on these. So, do we have an idea of how long ago these different traits showed up? And the answer is not really, okay? They're not intended to show the exact timing of divergence. And that exact timing is what's important. They do show relative timing, so what happened first, what happened second, but doesn't show exactly how many millions of years ago these different divergences happened. All right, so there are different types of phylogenetic trees, though, that can give you some clue as to the amount of time that's going on, but again, it's not exact. So there's this type of phylogenetic tree called a phylogram. Let's do my erasing here. Okay, that shows the number of genetic changes that have occurred. So the length of a branch corresponds how many genetic changes have occurred. And obviously, the longer amount of time that that organism has been around, the more genetic changes it could have collected. Okay, so in a way that kind of shows time, but again, it doesn't say exactly this is how many million years ago or whatever. So phylogram is a type of phylogenetic tree in which the branch length indicates the number of genetic changes that have taken place. Okay, here's an example of a phylogram. Just going to label this. You should probably draw yourself again just a simplified version of this. Okay, now we see we have one common ancestor down here for all these organisms, from Drosophila, which is a fruit fly, to a mouse and rat and a human. Okay, the most genetic changes that have accumulated are right here, right? Here's our longest branch. Drosophila has accumulated a lot of genetic changes since its common ancestor. If you look right here at the rat and the mouse, see we have a divergence right here. Since they diverged, there haven't been a whole lot of genetic changes. They have little short branches here. Okay, so they are very similar to that common ancestor there. However, they're very different from this common ancestor here because all of these branches represent genetic change to get to that point of a mouse or a rat. So hopefully that makes sense. Again, we don't know, millions of years or anything like that, but we do have an idea of how much genetic change has happened. Again, just a quick little outline of this phylogram is fine. You don't have to include all the pieces. Maybe you just want to do this section right in here from amphibian over to mouse, just to show that there's a difference in the amount of genetic change that's happened. Okay, there's another type of phylogenetic tree called an ultrametric tree. Again, this is all about timing. So it's the same as in a phylogram, so it's got kind of the same pattern in that each of the branches represents genetic change, but all of the branches are of equal length. So it's just arranged in a different way. It just sets it up so that they all end at the same point. And it's just easiest, again, to show this with a diagram. Again, you don't have to do the whole section of this in your notes, but maybe pick out that same section you did before with the amphibian over to the mouse, and you can see how across the top here, make my little dashes a little bit bigger so you can see, all across the top, all of these branches end at the same point. Right? Whereas the last one, the Drosophila, ended up a lot higher. It was like up here. And then the mouse and the rat ended up down here. Okay, so what they've done is they've just scaled it so that they've got all of the organisms ending at the same point because they are all current day organisms. Okay, this represents today. Okay, if we're talking about time, this is right now. We have these organisms present. But again, the branch length still represents the amount of genetic change. So Drosophila as that is very different from this ancestor down here, whereas the rat and the mouse are more similar to this creature down here. It's probably some sort of rodent that was the common ancestor of those two, whereas this thing millions of years ago down here at the bottom probably didn't look anything like a fruit fly or a fish or a rat or a mouse. I mean, there's been a lot of genetic change that's occurred since any of those existed. They do put like time periods on here, which is a little bit easier based on having that common ending point of today, but it's not the phylogenetic tree that tells us the timing. Okay, it's fossils that we've collected along the way that has given us that timing. But since we have it set up as an ultrametric tree, it's easier to put that timing on them because they're all on the same scale. Right? They're all ending in today. Okay, so just a couple of kind of wrap-up things of how we do these trees and really make sense of them. So in order to determine phylogenies, so make these phylogenetic trees, the scientists who do this, the systematists, have to think about or consider or look at the molecular evidence. Okay, so we're looking at mostly DNA, RNA, and proteins. Whereas if we were looking at doing taxonomy, like kingdom, phylum, class, order, species, all of that, you look at more of what they physically look like, their morphology. Okay, so this is a little bit more detailed, a little bit more precise way of classifying organisms, is this phylogenetic tree. Okay? And trying to figure out who's more related to who, this should make more sense if you have very little genetic change between two organisms, then that's probably the most likely to be the ones that are related, right? Those are considered to be the phylogenetic trees that are the most correct. Because we are going to assume that not a lot of genetic changes happen, that genetic changes happen at the same rate throughout time, throughout the millions of years. We haven't had like jumps of genetic change where all of a sudden a bunch of DNA changed and then it slowed down. Okay, so we can assume that the rate of genetic change is the same, and the phylogenetic trees that show that are the ones that are most correct. Again, I'm going to have a diagram to show you what I mean here. I always think that pictures help with understanding some of these more complex ideas here.